substrate binding to the enzyme? Penicillin binds to the enzyme. So it plugs up the active sites so the real substrate can't get in there and can't be acted on and can't be turned into peptidoglycan. So it interferes with the enzyme responsible for making peptidoglycan. So if you can't make peptidoglycan fast enough to keep pace with division, what's going to happen? Can't complete your transverse plate. When the two cells go to separate, they're going to have an open side and they're going to spill apart. So this figure is important not only for showing you transverse binary fission, but also for showing you the importance of this cross wall or this transverse plate. If peptidoglycan cannot be manufactured in pace with cell division, in its normal pace, then this wall is not going to be formed fast enough for the two cells to pull apart, and they're just going to have open sides and die. That's how penicillin works. Penicillin will stop the population from growing. Traditional penicillin is what we call a bacteriostatic antibiotic. It's a traditional antibiotic, and that's how they work. Whatever language you want to use. All right. So, remember we did this? Here's my body, homeostasis. Here's an infectious bacterium. Enters my body. They divide. They divide. They divide. They divide. They're growing, growing, growing. The population's growing. Here's my immune system. What the hell is going on over there? Maybe I should do something about it. Give me a minute. It's going to take my immune system up to 2 weeks to boot up. Meanwhile, I don't feel well. I go to the doctor, it gives me penicillin. Every time they go to divide, instead of dividing, they can't. And then the older ones die off. And then they go divide and they can't, and they die off. And they can't, and they die off. So the penicillin is bringing the infection under control by not letting it continue to successfully divide. Penicillin is going to act on the weakest bacteria first. More penicillin is going to get the stronger ones. More penicillin is going to get the even stronger ones. Doesn't all work at once. You have to take multiple doses because you're going to get the weak ones first. So it's knocked my population back to this now. But the strongest ones are what's left, so I got to finish my prescription. And also my immune system by now has booted up and will catch it and take over. That's how bacteriostatic antibiotics work. And that's what happens. It interferes with the bacterium's ability to form that transverse plate, so it's not going to have successful cell division. All right, so that's how penicillin works. It targets the peptidoglycan. You'll see other antibiotics also target the cell wall, the peptidoglycan. Antibiotics such as cephalosporins, bacitracin, even vancomycin. They inhibit synthesis of the cell wall. So, as you look at bacterial structures, there's going to be a target. That's the bacterial structure. And there's going to be a magic bullet. And that's the antibiotic. So for example, if the bacterial structure is the cell wall, specifically the peptidoglycan, the antibiotic is penicillin. Penicillin targets the cell wall. Let's put down a few other ones while we're at it. Cephalosporins will also target the cell wall synthesis, and also bacitracin and vancomycin. There are others also. Okay. All right, this chapter also talks about classification of bacteria. You already know this, and I think it's in another one of your PowerPoint presentations. But just so you know, here's your gram-negative bacteria. This is the new modern classification. Here's your gram-positive bacteria. Some have a high number of G's and C's in their genetic code, and some have a low number of G's and C's in their genetic code. And so the new classification is based on what? Genotype. The classification we do with our list is not based on genotype, it's based on phenotype. You know the difference? Here's your archaeobacteria. Here's some of your cyanobacteria probably in here. Why do I have to write? It's a different classification scheme. Here are some archaeobacteria. That's one of the hot springs, probably somewhere in the Wyoming, Yellowstone National Park. Couldn't think of it. Here's some other archaeobacteria. They're halophiles. They love salt. Characteristics of major groups of phototropic bacteria. Cyanobacteria. I don't expect you to memorize all of this. Just want you to know some bacteria live off of sulfur, so they're chemoautotrophs. Your cyanobacteria are photoautotrophs. They feed themselves by photosynthesis. These feed themselves with chemicals, maybe some photosynthesis also, because they have chlorophylls. We're not going to focus on those kind of bacteria. As long as you know what cyanobacteria are, we're good. Look familiar? Cyanobacteria. Here's some purple sulfur bacteria. Thank God we don't have to study those. Alrighty. This is the newer classification of bacteria. That's why I put this chart from your textbook here. Let me see where it's going. Notice how they classify the bacteria. Gram-negative bacteria, and we're going to do gram stains today in lab. Proteobacteria are gram-negative bacteria. Write that down right now, because it will be on all your tests. When we're talking gram-negative bacteria, what are they going to say? Oh, the proteobacteria. Oh, the alpha proteobacteria. Oh, the beta proteobacteria. What are we thinking? Gram-negative bacteria. So we're going to classify bacteria more for healthcare. This is more for, I guess, strict microbiologists. But you'll notice the same genera that you have in your list. Some of these bacteria you won't see in your list because I gave you a list for healthcare. This is the list with all kinds of bacteria. So, in the new tree of life, we learned about genotypes. For the new tree of life, what happened? we could sequence the DNA of bacteria. So they reclassified bacteria according to their genotype, their genes. Our way was the way Dr. Burgee did it, which was phenotype. What he could see in his microscope, what tests he could run on Petri plates and see the results in lab. So do you know the difference between genotype and phenotype? Genotype is your genetic makeup. And if they can look at the genetic makeup of every different bacterium that we can't find, they can classify them by how similar their genotypes are. Doesn't necessarily match up with phenotype all the time. Phenotype is how they look in your microscope and with different lab tests you do for their metabolism. Phenotype, if you ask me, is the better way for healthcare. Okay, here's another group in your proteobacteria, so we're still talking gram-negative. Notice they're glycolytic. What does that mean? Glyco, sugar, lysis, breakdown. They can break down sugars, and they're facultative anaerobes. Here's your Enterobacteriaceae. Gut bacteria. Entero means gut. Here's your Vibrios. Pasteuriliaceae. You're seeing a pestis is Pasteurella pestis. Here's more proteobacteria. Spirochetes, chlamydia. And here's your gram-positive bacteria. They call them firmicutes, actinobacteria. These names will seem familiar. Here's your Clostridium, Mycobacterium, Streptococcus. Okay, so we're going to classify our bacteria a little differently. Let me just look to the end of this. Let's see what this is. Mycoplasma.